All right, let's make it big. How you doing? This week we're going to come and talk about Ebola virus. And Ebola virus is a pretty neat virus. Let's go have a look. So here is Ebola virus. And these are three-dimensional images. And so we're actually taking slices through the virion right here. And so there you can see a couple little virions. There's, one, two, three, four of them coming off the cell. Here, each of those little gray tubes is the center of virion. And so this is from a technique called cryo-electron tomography. So let's rewind that and pause. Okay, so you can see this is what the virion looks like. It is a shape that is called filamentous. So there's a few, maybe one out of every 100, that'll have that sort of curly Q on the back. And that's a thing that's going to be repeated over and over because that's the famous image, the number one image. It just turns out that most Ebola viruses actually look like little sausages. They don't have the cool little curly Q, which is called the shepherd's crook. In the center, you can see a sort of a hollow space right there, and that is literally hollow. And then you've got the two sides over here and here of what is a tubular nucleoprotein. So it's a helix made of proteins and RNA. And so, here, I'll trace it out with my little thing. It'll be coiled up just like this, round and round, all the way until you get to the end. On the outside, it's got a matrix protein that's sticking the nucleoprotein to the membrane, and it's got a glycoprotein that's doing fusion. And you know what? That glycoprotein has all the same parts that you've come to know and love. So it has an outer part that binds the receptor, it has an inner part that unfolds and has a fusion peptide and a fusion motor, and it just works the same way as coronavirus or influenza virus or HIV, even though the protein itself is basically unrelated to those things. So let's watch it a little bit more. And there we go. Got a little moving image in here. I think that's quite nice. And right toward the end there, as you're coming up through the surface, here we go. There we go. These little black dots, there, those are the glycoproteins on the outside. And so they're just nicely arranged glycoproteins there. We'll see some artists' renditions of Ebola virus later on in the talk, and you can see how they look, how they compare. So this one I like. This one's by a guy named David Goodsell, who I think just does the best biological animations. And so here, what we have is the glycoprotein. And so the bit that's carved out, like down here, that doesn't really look like a protein, that's the bit we haven't solved yet. We know there's three copies there together, and we know that there's a domain on the outside that has not yet been solved, and there's a domain at the bottom. This would be the equivalent of GP41 or S2 or HA2. Yeah, the bottom bit that unfolds and does the fusion. We got a matrix protein, and it's going to form that matrix-like layer, which, remember, a matrix layer is a layer that kind of underpins the membrane. So here's our membrane, this sort of pinky thing right here. The matrix layer is going to sit right underneath that and reach up, and the matrix layer is going to bend it into its proper shape. And the matrix is also going to interact with, there you go, the tails of the glycoprotein. And so even though this is an artist's rendition, it's a pretty darn accurate artist's rendition. Here's the nucleoprotein. We know it makes that sort of hollow helix in the center that you can see it. And then there's a couple other minor proteins, this VP35, 30, and 24. And we'll see that some of those are going to be involved in immune modulation, essentially shutting down that RIG-I and MDA5 pathway. The polymerase is also going to be packaged because this is another negative-stranded RNA virus. So it's going to come in with a polymerase and with a backwards genome. Now the genome is going to be one single piece of negative-stranded RNA, but it's hooked up kind of the same way as coronavirus. So it's got the big polymerase gene first, and it's got all the other genes in order. And like a coronavirus, it is going to make subgenomic RNAs, and it's going to express each of these genes so that one gene is expressed from each RNA. The only difference is that this thing doesn't put a leader onto its messenger RNAs the way coronavirus does. It just makes shorter messenger RNAs that stop at the TRS, and so they go from the TRS all the way to the end, and you'll have one of those for every TRS that goes along. So first, second, third TRS, all the way to the end. There you go. Yeah, vaccine efforts so far tend to be focused just on this outer protein, which is the glycoprotein. That seems to be the big one that you get a lot of. That's the only one that the immune system can see because it's the only thing that's sticking out of the outside of the virus. The other little purple proteins that are all different shapes that you see sticking up here are all host proteins, like proteins from the host cell that just accidentally got incorporated. So that's what they look like. All right. So there is a cellular protein called TIM1, Tim1, and it is the one that acts as a receptor for Ebola virus. And Marburg virus is another one of these Ebola-like viruses. So they're both in the genus Ebola virus, if I remember correctly. Marburg virus is one that they initially thought was spreading via the air, but since then it hasn't really spread at the same rate as if it spread through the air. And so we think that may have just been a mistake in the initial things. But yeah, can you imagine airborne Ebola? Yeah, that's Marburg. That's the thing that virologists worry about anyway. Because Ebola has a fatality rate in a good case of about 50%. And in a bad case, it'll be around 90% fatality rate. Compare that to any other virus that you know, and it stacks up as being pretty bad. So, yeah, the Ebola virus glycoprotein is going to attach to TIM1, and then it's going to go through that similar set of unfolding metastable states, and it's got to be cleaved right there at the top of the fusion peptide, and that is how, yeah, that's how it's going to work. All right, interesting things. So these first two little slide sets are going to be sort of interesting Ebola facts to kind of get you in the right place so you know what we're talking about and, yeah, just give you something neat that you can tell people about. So actual Ebola virus is going to be found across the center of Africa and shown here in panel A. So this is all the places where we know that the hosts of Marburg virus can grow. And let's see, panel D is where all the hosts of Ebola virus can grow. So far, Ebola is only found in just about this brown band running across here. You see it's going through the top of the Democratic Republic of Congo and going through the Republic of Congo as well, kind of that area of the world. You tend to get Marburg over here by Kenya. You tend to get more Ebola over here. And that giant outbreak in 2013 or maybe even 2012, I forget when it was over here weirdly on the west coast of Africa. So that was quite unusual. But what you see here are hosts. So the hosts of all these Ebola-like viruses are thought to be bats. Once again, bats. But this time it's megabats, these sort of giant fluffy bats. They mostly are fruit bats, like to eat fruit. And as you can see, the bats live all over the place. So Marburg virus is only found here, but there are bats all the way down in South Africa or on the southern tip of the Arabian Peninsula or a little bit of Turkey right there. And those bats could carry the virus. They're the same species. So if they have contact with bats from here that have the virus, then that could happen. Down here, regular Ebola virus, again, only found for the most part in this part, sort of central to Western Africa, is where it tends to be. Occasionally you'll get a case over here in Kenya. But there are bats that definitely can carry Ebola virus, and they go all the way down to the bottom of Africa, up to the southern coast of Turkey, which is part of the European Union, so that's great. And there are also, look at that, bats that could carry it all over sort of southern Asia, including parts of China. So yeah, that's terrible. But even neater, there are two other Ebola viruses. One's called the Reston Ebola virus, that's in B, and the other one is called the Yoviu, I believe it's Spanish, virus. And these both have all the same genes as Ebola virus, the one that kills you, but neither one of these is associated with a single human death yet. So you've got two viruses here with about 50% and 90% maximum kill rates, and you've got two viruses here that have all the same proteins that have 0% fatality rate. So there are differences between these things, and the differences are not completely understood. There are lots of genetic differences, small differences, but they have all the same proteins. So here, look at that. Look at that cute bat. That is one of the gorgeous little bats that carries Ebola. It's a little, they call it a flying fox. And they're biggish. They're like pigeon size. Whereas the bats that carry the SARS coronavirus are tiny. They're about maybe 2 inches tall. These guys are much bigger. So, all right. So this is the breakdown. Oh, here we go. It was, yeah, that's right, 2014 when the outbreak was declared. We think the first case, though, may have been back either in January 2014 or in late 2013. Just took a little while to get going. And so this most recent giant outbreak spread in Guinea is where it started. But it was kind of at the border between Guinea and Liberia and Sierra Leone, which are all over there in West Africa. And interestingly, Guinea was a French colony, and so the French government went in there to do to provide doctors and hospitals and things like that. Sierra Leone was a British colony, and so the British did that one. And Liberia started out as an American colony. This is a place where they were planning to repatriate freed slaves after the Civil War. They just went over and, I don't know if they conquered or just bought, like a country in Africa, to try and put people. Not many ended up going there. And of course, there were already people living there. That's the problem with all these narratives of discovery, isn't it? You find a place, but there are people already there. But it's okay. You got a flag. Anyway, yeah. So this is how it went, and it was bad. So these are weekly reported cases, and at the peak, we were getting about 500 weekly reported cases. And so of those, you'd have about half as many deaths to 60% as many deaths. So think like, I don't know, 200 to 300 deaths in this per country in the really badly affected countries. Compare that to the coronavirus, and we have far more cases, but not as many deaths. So I guess that's all right. Okay, so another fun fact about Ebola virus. It turns out that the virus is a lot more persistent than anybody originally knew. And so they figured out that the virus can stick around in a couple of places. So it lives inside the milk ducts, actually inside breast milk for a little while. It can live inside the testes for a considerable amount of time. And it turns out it can also live inside the human eyeball. Yeah. And we have cases of each one of these things. And yeah, 2 of those have led to infection. So far, we haven't had a confirmed eyeball related infection yet. I don't know what that would even look like, but theoretically possible. And so this is saying that of people that had the virus and then get over the virus and are negative by RT-PCR, which is a way to count copies of RNA, and it's the same sort of test they do to detect coronavirus now. The Ebola virus RNA was detected in semen of everybody in the study 2 to 3 months after they got after onset of Ebola virus disease. So Ebola also lasts about a month. So this is 1 to 2 months after they were clear, and in about 65%, 4 to 6 months after onset. So the virus is hanging out for 5 months, and plenty that were 9 months after getting the disease. And one participant who, was at 10 months, probably had a little bit there, but it was at around the threshold of detection. So there have been cases, definitely, where men would go out after they had gotten over Ebola and, try and lead their normal lives and you end up with sexually transmitted Ebola, which is a heck of a thing. So here's the one example of the other thing. This is Dr. Crozier. He was one of the U.S. doctors that went over to Liberia to help treat people there. While he was in country, he got infected. There were a couple cases like this where it was some fault in the protective clothing or probably in the way that it was taken on and off. But the conditions are just terrible in these field hospitals over there. and it was way over 100 degrees every day while they were trying to do this. And so, yeah, they did a great job. But the Ebola virus got into him. He got sick. He got better. But then this happened to his eye. Look at that. And so now he has a permanently dilated eye that is actually a different color to his other eye. And they probed it and found out that this is Ebola virus positive. And so we're not sure if he's going to carry Ebola virus for his entire life or just for a long, long time. But yeah, he was still positive something like a year after overcoming Ebola. So that's one of the things the virus does. It's kind of like coronavirus in that way. It just sort of hangs around. But it's not like HIV. It's not actually copying itself into the genome. It's just really good at the old game of hide and seek. All right, that's the end of the first Ebola section. Then we're going to go on to the second one. So I hope you enjoyed that. Oh, do we have 2 more slides? Yeah, okay, we have 3 more slides. Let's do our 3 more slides. Then we'll be done. Okay, so here was the big outbreak. Now, since then, there's been another outbreak, and this thing actually went on and became the second biggest outbreak. It was over in the Republic of Congo for the most part. It is nearly contained. Last I checked, it had been about 20 days since they had a case in most of the towns. I think there might have been 1 or 2 active cases still in one city, but it does seem to be getting close to under control. But yeah, take your eye off it, and Ebola comes back, or comes back from people who have gotten over it but not quite gotten rid of it. So these were the areas that were infected. This is the total number of cases that were reported, and you were able to check these in real time. Very nice World Health Organization briefings on that and on the current Ebola outbreak. So it's worth looking at. Of course, people will be people, yeah, as it says here. And as you're listening to people, to stories of people and what they're doing in response to the coronavirus outbreak and some of the crazy stuff that people get caught doing, consider this. Yeah, it's nothing unique, nothing new. So in this case, we're talking about some people who had actually died of this, and so these people died. There were high-risk contacts. Yep, and all 5 cases were epidemiologically linked. Great. All 3 probable cases died within a particular window and were not buried safely. So what was happening then is that relatives were coming in and taking away the bodies of people who died or refusing to bring them to the hospital because Ebola remains infectious and it gets all over the surface of the skin. It actually comes out in the sweat in the last days of life. And so that is one virus where, yeah, touching somebody and then rubbing your eye, nose, or mouth can actually lead to Ebola in that case. For something like a coronavirus or a flu virus, somebody would have to cough on a surface, and you generally don't get as much virus that way either. It's a relatively low amount. But yeah, and so they checked it out, and each one of these unsafe funeral events then goes on to spread. And it ends up spreading to something like an average of 40 people per funeral. And so funerals were terrible, but people being people still wanted to have them. And so, yeah. This is the way they handled it. Look at the way that other people are handling coronavirus, and yeah, they would like life to go on, but there's some you just can't reason with a virus. Yeah, there's some things you can't do. The other interesting thing about the Ebola, and this is the last slide in this section, is that you would be able to tell who is going to make it or not make it fairly early in the symptomatic phase. So Ebola also has about a 7-day lag between when you would get infected and when you would start to show symptoms. It's something like 3 to 10 days, but pretty similar to coronavirus. And then you can see here the darker sort of gray bars are the amount of virus in people who had fatal cases, and the white bars are the amount of virus in people that had non-fatal cases, so the ones who survived. And so you can see the difference, right? The gray bars are up here, and they're generally about 1 to 2 logs. So 1 log is 10 times higher, 2 logs is 100 times higher, compared to the ones that survived. Now, these are all people who are not getting any specific medication. because this all comes from before the medications. Well, actually, there still are no medications, but this was before the vaccine was even available. And so there was nothing but bed rest and IV fluids because Ebola gives you a lot of diarrhea, and that can drain you, and your organs will just shut down if you get too dehydrated. But yeah, there's definitely a genetic difference that led some people to control the virus without drugs and other people to not control the virus. And the big thrust of this talk is going to be looking at that genetic difference. What is it? How does it work? And yeah, I think it's a really neat story that applies to a lot of other viruses as well. So let's go on to that. And that's the end of this part of the lecture.